Good afternoon, students. I hope you're all good. Now, today we'll start with the next subtopic of our chapter Human Reproduction. What we are going to start with, the topic is the structure of the spermatozoon. Here on your screens, you can see the diagram of a spermatozoon. It is quite important from examination point of view. Now, if we look at it, you can see that a spermatozoon consists of four parts: head, neck, middle piece, and tail. They are marked on the diagram, which you can see. Now, the head is the topmost part. You see, it is a flat and oval in the human sperm. It is composed of a large posterior nucleus and a small anterior acrosome. The nucleus consists only of condensed DNA and basic proteins. Acrosome is formed from the Golgi complex. It contains hydrolytic enzymes and is used to contact and penetrate the egg in fertilization. Its use we'll see when we study fertilization. Okay. So the next part, which is neck. The neck is very short and contains two centrioles lying one behind and at right angles to the other. The proximal centriole plays a role in the first cleavage of the zygote. A distal centriole gives rise to the axial filament of the sperm. Then comes the middle piece. The middle piece is cylindrical in the human sperm. It contains many mitochondria tightly coiled around the axial filament. The mitochondria provide energy for the movement of the sperm in the female's genital tract. Middle piece is also termed as the powerhouse of the spermatozoon. Why? Because the amount of energy which is provided is from this middle piece and it is limited. If a sperm fails to contact an ovum within a specific period, which is around 24 to 48 hours, it exhausts its energy and dies. Last part of it is the tail. The tail is very long, slender and tapering and is formed of cytoplasm. A fine thread, the axial filament, arises from the posterior centriole and traverses the middle piece and the tail. It may project freely behind the tail as an end piece. The mammalian sperm has an additional outer ring of 9 coarse accessory fibers. The entire sperm is enclosed by a plasma membrane. The spermatozoa swim about by vibrating their tail in a fluid medium in search of ova. Okay, right now, so you have understood the structure of spermatozoon, which is divided into four parts. Now next we'll take the structure of the female gamete, that is the structure of the ovum. So I have displayed here two diagrams of ovum. Here you can see the two layers, that is the corona radiata and corona pellucida, and then you can see the ovum. And in the next diagram also you can see here you can see the sperms also arriving near the ovum for fertilization, and you can see the different layers here also. The corona radiata, here are the cells of the corona radiata, then zona pellucida, the perivitelline space also you can see, and the ovum also you can see. Right, so both these diagrams are of the human ovum. Now the ovum, as you all see here on the screen, is a rounded non-motile cell. It is much larger than most of the body cells. Its size is around 0.1 mm, whereas the other body cells, they come under 0.01 mm. It is almost free of yolk, so it is also known as alecithal. It has abundant cytoplasm, which is known as ooplasm. It has a large nucleus termed germinal vesicle and a prominent nucleolus. at the center and surrounded by plasma membrane, which is also known as oolemma. The latter gives off microvilli to absorb food material from the follicular cells during the growth. The tiny vesicles known as the cortical granules lie just under the egg's plasma membrane. There are no centrioles in the ovum. So this is very important, that there are no centrioles in the ovum. The ovum shows polarity. Its side which extrudes polar bodies is called animal pole, and the opposite side to this is known as vegetal pole. The ovum is enclosed by two adjacent egg coats, inner layer which is thin, transparent, non-cellular zona pellucida, and an outer thick corona radiata, which I showed you in the diagram. So the outermost is the thick corona radiata. The zona pellucida is secreted by the ovum itself and is also known as the primary egg membrane. It is composed of 3 different glycoproteins. The corona radiata is formed of radially elongated follicular cells. It is the secondary egg membrane. And there is a space in between which is known as the perivitelline space. So have you seen this diagram now? So now we move to the differentiation of the gametes. How these gametes they differentiate, the spermatozoon as well as the ovum. So first we'll take up the spermatozoon and we'll come back to the diagram of the spermatozoon. So the transformation of a spermatid into a spermatozoon, it involves certain changes, and these changes are change in the nucleus. The nucleus gradually becomes smaller but longer, DNA condenses, RNA and nucleolus are lost, chromatin fibers become closely packed. Then there are changes in the Golgi apparatus. A small part of it forms the extrosome that joins the interior end of the nucleus. The rest of the Golgi apparatus is discarded along with most of the cytoplasm. Then there are changes in the centrioles. The centrioles shift behind the nucleus and come to lie one behind the other. The anterior or the proximal centriole orients itself at a right angle to the long axis of the sperm in a depression in the nucleus. The posterior or distal centriole orients itself parallel to the main axis of the sperm. This gives rise to the axial filament of the sperm. Remember, we have studied it just now in the structure of the spermatozoon. Then there are certain changes in the mitochondria. The mitochondria shift behind the centrioles and get arranged in a spiral around the axial filament. And the changes in the cytoplasm. The cytoplasm extends around the entire sperm as a thin sheet. Much of it is not used and is discarded to make the sperm lighter. And the changes in the plasma membrane. The plasma membrane extends around the developing spermatozoon to cover it fully except at the end piece. Now we come down to the changes in the ovum. Now for the differentiation of ovum, there is a change of an oocyte into an ovum, which involves the change in the nucleus and change in the cytoplasm. The nucleus enlarges due to increase in nuclear sap. This facilitates extension of chromosomes for transcription. Nucleolus also enlarges. The cytoplasm increases in amount and is called ooplasm. Ribosomes and mitochondria increase in number. Endoplasmic reticulum becomes more elaborate. Golgi bodies accumulate and give rise to cortical granules that gather in the peripheral region of the cytoplasm. Cytoplasm becomes rich in RNA, ATP, enzymes, and food materials. All this makes the ovum much larger than the somatic cells. So, is it clear to you? The difference also and the different changes which occur for the differentiation of the spermatozoon and the ovum. Now the next subtopic which we will take up is the menstrual cycle. Here you can see this is the schematic representation of the menstrual cycle, which shows the different phases, the days involved, the level of hormones. All these things you can see here displayed on your screen. So these are all the events, that is the uterine events, the hormonal levels, the ovarian events, the pituitary hormone levels, everything in one graph. So please listen carefully about the different stages and the changes which occur during the menstrual cycle. Now the ovum or the female gamete, its formation in the females is a cyclic activity and it takes about 28 or 29 days and involves changes in the structure and function of the entire reproductive system. This is what is known as the menstrual cycle. The menstrual cycle occurs only in the primates like monkeys, apes, human beings. The first menstruation begins at puberty and this is known as menarche. The menstrual cycle in humans lasts for around 1 month, that is 28 to 29 days to be more exact. The days are numbered from the first day of the blood flow in the menstrual period. In other words, the cycle of events starting from the first day of menstruation to the start of the next menstruation is known as one menstrual cycle. In human females, menstrual cycle ceases around the age of 50 years and this is known as menopause. So here there are two terms, that is, one is the menarche and the other is the menopause. We can divide the menstrual cycle into three different phases, like the follicular or the proliferative phase, second the luteal or the secretory phase, and the menstrual or the bleeding phase. Now if we take up the follicular phase, which is also known as the proliferating phase, this phase lasts for around 14 days. It comprises some events like the anterior pituitary lobe secretes FSH. So here the levels of FSH, you see they start rising. Initially, the FSH stimulates the growth of the selected primary ovarian follicular and maturation of the primary oocyte in this follicle. The FSH also stimulates the follicle cells to secrete estrogen, which is a female sex hormone. The estradiol inhibits the secretion of FSH and stimulates the secretion of LH, that is the luteinizing hormone, from the interior pituitary. The LH induces the mature Graafian follicle to burst and eject its egg into the fallopian tube. This is known as ovulation. It occurs around 14th day. This peak is also the LH surge. The estradiol also stimulates the uterus to prepare for the implantation and nourishment of the fetus likely to arrive after ovulation. The wall of the uterus, relatively thin in the beginning of the cycle, progressively becomes thick by greater development of its smooth muscle layer, that is the myometrium, the proliferation of its epithelial lining, the endometrium. The blood vessels in the endometrium, they become long and coiled. Glands develop which secrete a nutritive fluid into the uterine cavity. Under the influence of estradiol, the lining of the fallopian tubes is thickened and its cilia and their movements, they are increased. These changes prepare the fallopian tubes for conveying the released ovum into the uterus. Now the next phase is the luteal or the secretory phase. This phase lasts for around 10 days. It involves the following events. The peak of LH together with prolactin hormone from the anterior pituitary lobe stimulates the follicular cells of the empty Graafian follicle to form a yellow body which is known as corpus luteum. The corpus luteum secretes another ovarian hormone called progesterone and a smaller amount of estradiol. The progesterone and estradiol continue the hypertrophy of endometrial lining in the uterus and fallopian tube and maintain it throughout the pregnancy. The progesterone is necessary for the proper implantation of the fetus in the uterine wall. It stimulates the endometrial glands to secrete a nutritive fluid for the fetus. So, this phase is known as the secretory phase. The progesterone inhibits the release of FSH so that it may not develop additional follicles and eggs. Is that clear? Then we come down to the next phase, that is the menstrual or the bleeding phase. The phase lasts for about 4 days. It involves certain events, that is, in case there is no fertilization, the secondary oocyte undergoes autolysis and the progesterone inhibits the release of LH from pituitary. Reduction of LH levels causes regression of the corpus luteum by autolysis and a consequent fall in the progesterone level in the blood. The uterine lining dies due to deficiency of progesterone and is sloughed off. Blood vessels rupture, they cause bleeding. The lining of the fallopian tubes breaks down. They cast off uterine and fallopian tissue. The blood that oozes from the ruptured endometrial blood vessels and tissue fluid from the endometrial surface passes out through the vaginal opening. This is known as the menstruation or the menstrual flow. It occurs after about 25 days and continues for around 3 to 5 days. Menstruation is often described as the funeral of an unfertilized egg, or it is also known as the weeping of the uterus for the lost ovum. The basal part of the endometrium remains intact during menstruation and is the source of the new uterine lining and glands. The lowered levels of progesterone and estradiol due to the regression of corpus luteum cause release of FSH from the interior pituitary. This initiates a new cycle. So here in the next diagram also you can see in a cyclic way it is shown that the cycle begins from the first day of the menstrual phase. Then it is continued around up to 4th or 5th day. Then the time is of the post-menstrual phase, the proliferative phase, and in it here you see there is ovulation around the 14th day of the cycle, and then this proliferative phase enters into the secretory phase, and here the corpus luteum begins to degenerate. And then in case there is no fertilization after 28 days, then the next cycle will start. Now the effect of fertilization. If fertilization occurs and fetus is implanted in the endometrium, the trophoblast cells of the developing placenta secrete a hormone called as human chorionic gonadotropin HCG. This hormone, like luteinizing hormone, maintains the corpus luteum and the secretion of progesterone and estradiol by it. These two hormones check the breakdown of the endometrium of the uterus. The absence of menstrual bleeding is the earliest sign of pregnancy. By the 16th week of pregnancy, the placenta produces enough progesterone and estradiol for a normal pregnancy, and then the unnecessary corpus luteum undergoes shrinkage. Right? So, in the case of miscarriage, what happens? Premature degeneration of corpus luteum. This is the common cause of miscarriage in the earlier weeks, right around from 10 to 12 weeks of pregnancy. Now, during pregnancy, the HCG is detected in the urine, and this forms the earliest signs of detection of the pregnancy. If these changes, if the fertilization does not take place, then the progesterone levels, they will come down and the estrogen levels will again start rising, the FSH level will rise, and the next menstrual cycle will start. But in the case of man, there are no cyclic changes in the reproductive organs. A male can produce the sperms throughout the life once attaining puberty. This is all for today's lecture. I hope it was clear to you. Any doubts, please you can ask me. Thank you.